Thank you for attending to this webinar about microfluidics applied to soil ecotoxicology and microbiology. It is brought to you from the European project EDAPHOS by one of the partners, which is us, the Microfluidic Innovation Center. Today, you will have not one, but three presentators. Jesus Dominguez, which is a third-year PhD student at Gothenburg University, doing his thesis in collaboration with the MIC. So, hi, Jesus.
Hi.
And there's also Olivier Fournier and myself, Evelyne Mancel, who are research engineers at the MIC. So this presentation will last about 50 minutes with a Q&A session afterward. Do not hesitate to add your questions in the chat during the presentation if you have them. And also, the presentation is recorded. Tell us if you have any problems with that. But the Q&A session will not be recorded, so it's a totally safe space. Any questions are welcome. And it will begin by answering the question of what is microfluidics. Then Jesus Dominguez will present you his research work about microfluidics applied to microbiology and soil studies. And Olivier will finish the presentation by exploring innovation in microfluidic. So firstly, let's talk briefly about what's the MIC. Well, MIC's operations are based on working as closely as possible to the market in order to develop innovative products tailored to current research needs. So that works in particular through collaborations on European projects, which is a theme in which the MIC is practically successful since we have been granted more than 50 projects with a high success rate in submissions. So now to attack the core of this presentation, what's microfluidics and what's the physics behind it? Well, the story of microfluidics starts with the beginning of micro and nano technologies. So that's the idea behind Feynman's speech that he had been given in Caltech in 1959. And it is entitled "There's Plenty of Room at the Bottom." And the idea is that if we succeeded making really small devices, then it could increase the throughput, reduce the costs, and open the door for parallelization. So that happened in the 18th with the invention of micro-electromechanical systems, which is the miniaturization of all kinds of systems. And when you adapt it to fluid flows, then it's called micro-total analysis system or lab on a chip. And since the size of the flow is adapted to the dimension of the system under study, well, let's say cells, for instance, then the interaction becomes more efficient. It also reduces the need in sample volume, reduces the required total analysis time, and the overall apparatus becomes compact and portable. So what we called the footprint. So that development led to the creation of a new discipline, which is called microfluidics. And what is it by definition? Well, if we want to quote the words of Whiteside, it's handling of fluids in technical apparatus having internal dimension in the range of micrometers up to a few millimeters. So basically, it can be defined as the science and technology of manipulating extremely small volume of fluid within a network of miniaturized channels. If you want to quantify all of that, well, the channel width is under hundreds of micrometers. The channel heights could be up to 100 micrometers as well. The flow rates would be from microliters per minute up to a few milliliters per minute, and the volume from picoliters to milliliters. And to try to have the ideas a bit more clear about it, we can take the example of a really basic setup, who would be a pressure controller driven by a computer, which send air to a liquid reservoir, and that pressurization sends a liquid in the microfluidic circuit, which is mainly a chip containing the system to be studied. And you can add a flow sensor to adapt the flow rate and an end reservoir for collection or waste. Now, this last definition is a framework in which the systems are made by human beings, and it could sound quite restrictive, since nature also produces astonishing micrometrics system that include the control of flow circulation. So here's the example of trees, which possess a complex network of capillaries that allows to supply water homogeneously from the ground to the leaves, with flows in micro and even sub-micro channels. Now, if we go back to human activities, we can ask ourselves, what's the application of microfluidics? Well, thanks to local sensor and retractions electronics, microfluidics are able to finely regulate fluid flow conditions, including pressure, flow rate, temperature, so gas exchanges. If we take the example of O2, you could put your sample in condition of hypoxia or anoxia, for instance. And another example would be to be able to properly regulate the pH. It also allows for miniaturization of multiple assays and tests, as well as the automatization of the overall process. So such precise control of the environmental parameters can be used in many different fields. And the first we could think of is microfluidics for biomedical application. We can take the example of cell analysis that greatly benefits from microfluidic perfusion with the possibility of generating finely tuned gradients of concentration or of shear stress, etc. And also the possibility to perform live cell imaging thanks to transparent chip. It is also true for more complex systems such as organ on chip. What we call organ on chip is a multi-channel 3D microfluidic cell culture, which is an integrated circuit that simulates the activity, the mechanism, and the physiological response of the key theater of an organ. And it does constitute the subject matter of significant biomedical engineer research. Another example of biomedical application which greatly benefits from microfluidics is point of care, which is medical analysis performed directly in the patient environment. So it is already used for pregnancy testing, for HIV diagnosis, for glucose biosensing, and screening of drug abuse. There's also many examples of microfluidics application outside the biomedical area. And the two next slides, we will shed light on droplets and soil on a chip, which is two applications that we think could be really interesting applied to natural media studies. So first one is droplets. Well, what's droplets? You might ask. Well, unless continuous flow system, droplets use non-miscible phases to create slugs or discrete volumes. And they are used basically as microreactors containing different chemical or biological compounds, allowing massive numbers of independent reactions to be performed really quickly using a minimal amount of total reagent. Indeed, each microreactors can be individually controlled simply by tuning the ratio between the two non-miscible phases, either by pressure or by flow. So these microreactors can be individually controlled, transported, mixed, and analyzed. And it's also possible to make multiple identical microreactors units in a short time, thus easily achieving parallel processing and allowing large data sets to be acquired really efficiently. And one thing that could be added is that due to high surface to volume ratio at the microscale, heat and mass transfer times, as well as diffusion distances, are shorter, accessing way faster reaction times. So that would be the first example of like non-biomedical application, but there's also what I think could be really interesting for your research, which is soil on a chip, which relies on the ability to confine organisms within a defined microenvironment in a really precise and robust manner. So this allows the experimentalists to address specific parts of an organism with really fine spatial and temporal resolution. That could be obtained resorting to chip engineering, for instance, or gradient generation. It allows organism growth and distribution to be managed with ease, and it's also compatible with a great variety of organisms, which allows co-cultures and studying the interaction between the different organisms. So here's the illustration of an article from EDAPHOS Hammer's group of soil organism and processes recorded in a microengineered chip, which was colonized in situ when buried in the field. Now, if you want to explore a bit more about microfluidics and soils, then in contrast to biomedical and drug discovery fields, conventional soils testing is yet limited by the lack of biocompatible automation, the lack of integrated optoelectronic sensor, and the lack of analysis algorithm. And microfluidics would allow measurement of organism viability, of course, with a high accuracy, in high throughput, and with high integrated consistent analysis. Also, it would allow for both lab tests and real-time environmental monitoring application. So these new methods should at the same time minimize the use of test animals, reduce the cost and time of testing, and ultimately improve the ability to identify toxicity hazards and support, as you might have heard of, the new legislative initiatives. Now, if you want to go a little bit deeper about all the advantages of microfluidics applied to soil studies, then you have, let's say, 4 big branches of advantages. First would be the parameters control, as we already mentioned before, with the possibility to mimic outdoor environment thanks to hypoxia and range of temperature. For instance, you could also tune the nutrients and the cultures in your chip. And for concrete research example, here's an article from 2015 where the control of the pH in a microfluidic device allows to separate E. coli from humic acid in order to understand better their interaction. A second big advantage of using microfluidics would be spatial evolution. So this article describes how microfluidics can be used to mimic the soil environment. Indeed, arenas for the study of microbial physiology, behavior, and interaction can be fabricated, allowing precise control over where and when microbes enter the system, with the possibility to restrict the encounters to few individual cells, for example. The third axis would be temporal evolution. So for a more concrete research example, here's an article that would be further discussed by Jesus, which shows how microfluidics can monitor the growth of microbial culture, accessing the different phase of their life cycle with high temporal resolution. And last but not least, microfluidics allows you to study a very diverse quantities of organism. And here's a quite exhaustive 2000 review of the different research work in microfluidics applied to ecotoxicity, which shows how it can be used on unicellular organisms, so bacteria, protozoa, and yeast, but also on microalgae, nematodes, crustaceans, and even many more organisms. So that would be a broad introduction, and now Jesus Dominguez is going to talk to us through his research work, which is applying microfluidics to microbiology in cell study. Let me just give the hand to Jesus. Yes.
Yes, perfect. Thank you very much. So yes, I'm Jesus Dominguez, and I'm a PhD student in Gothenburg University, and now we'll talk how we implemented, well, all of these concepts that you've heard before in my research, especially collaborating with the MIC. So for that, I will be talking mostly about bacteria culture, but this applies, and there's going to be a perspective also in soil studies in general. So yes, first of all, let me see if I can, oh, next. Oh yeah, oh, sorry. So the first thing is that when we try to study microbiology, we're limited by traditional methods. And this includes the use of, for example, Petri dishes or falcon tubes, which are macroscopic objects out of the range in which these microorganisms actually form and developed. And then if you want to observe how things are happening more intimately, you're going to have to manipulate and take a sample from it. So this means that it's ex situ observation. You're not observing the actual system that you want to know. You're observing something that it was extracted, that is not dynamic anymore. And that actually requires a lot of manual work, which is, for example, like pipetting, and also includes striking, etc., which at the same time yields a very low parallelization. You will have a limited amount of samples that you can do yourself, and you have to do manually, that also have a restricted control of conditions. So in this plate, the conditions are going to be mostly uniform, and you cannot change them over time or change them to your liking to have different type of areas. So this overall makes that a lack of automation and integration of this kind of process makes a very inefficient use of your time. So for that, then I want to talk a little bit about what we're trying to do. That is, we're trying to mimic environments that are quite complex. And the main example of this is going to be soil. Soil is an environment characterized for its heterogeneity. And this heterogeneity comes in different levels. So first we have physical heterogeneity. So you have first microaggregates that come together in macroaggregates by joining because of fungal hyphae or, for example, plants' roots. And they have different properties. So some of them are going to be more permeable, different porosity, etc. And this is going to be linked then to the chemical heterogeneity. So not only these microaggregates can have a different composition, that means some organic materials or inorganic and mineral materials, but also because of their different physical properties, some of them are going to be, there's going to be a different range of accessibility, for example, to gases or to water because of the permeability. And this is going to make an environment that it's highly different, and there's going to be a lot of species that are adapted to each part of that. But these species are not isolated. They actually interact with each other. And sometimes this is a crucial part of their survival. So there can be different synergetic methodological paths, so there can be a lot of type of relationships, such as predation, mutualism, commensalism, etc., that are important for their survival. So what we want to do is to replicate these kind of environments, and microfluidics gives us some tools to do that. So we want to talk about physical heterogeneity. As we said before, we have soft lithography and ways that we can have very tailored and specific accurate shapes that try to replicate the soils. but it doesn't need to be as complicated. And we can introduce, for example, microparticles that actually will be randomly distributed and making cavities and channels that can give a better approximation to what the soil is like. Then in terms of chemical heterogeneity, we're gonna be able to do, for example, thanks to the laminar flow of microfluidic systems, we're gonna be able to implement chemical gradients. And this is gonna make that we have different types of chemical accessibility in different regions. So for example, this can be interesting for oxygen or water access. And then finally, we're gonna have the biological component. So it's easier to do in microfluidics co-cultures because you can see how these species interact with each other in contact. But sometimes we don't even want them to be in contact. We just want them to be in contact with their metabolites. And that can actually be done through, for example, the use of membranes or through the use of nanochannels that don't allow the passage of bacteria, but the substance that they produce, they can actually be in contact and be exchanged. So with all of this, what is going to be our goal? Well, it's to adapt and validate microfluidic platforms for in situ bacterial behavior observation. So for that, we want direct and long-term observations. We want to have a high temporal and spatial resolution in a controlled microenvironment. So we want to be able to control how things are happening dynamically. And also specifically, we want meaningful throughput. We want to be able to use efficiently our time. So for that, I'm going to start with the first of the projects. That is an adaptation of the AIMS genotoxicity test. First of all, genotoxicity is the capability of a material to induce changes in the genetic material of an organism. That means induce mutations. So usually for this test, a specific strain that is genetically modified, in this case Salmonella, to not produce histidine, a necessary amino acid for the proliferation. So they cannot survive in environments that do not have available histidine. So when exposed to a non-genotoxic material like water, we're going to have low proliferation of the strain. However, the situation is going to be different if we have a material that actually enhances and changes, can affect the genetical material. So some of these times, they're going to produce mutations that can revert back the bacteria to a state in which it can actually produce histidine. This is going to produce a high proliferation of the bacteria. So under this frame, what we want to do is test whether or not a sample is genotoxic or a material is genotoxic or not by using and profiting microfluidics and bacteria. So for that, we implemented this basic design. So we have 3 different lines that are going to be the positive control, negative control, and the test sample. And we also divide them in 3 wells because 3 is usually a staple for for the number of replicates of a biological experiment. So for that, also here, I wanted to draw a little bit of a comparison. So I'm comparing, for example, using agroplates, that is purely traditional, using microplates, that would be the commercially available solution that there is, and then miniaturized, that it's the platform that we developed. So in terms of time, we reduce the time we need to perform a full experiment, also the reagent volume that you need of the test sample, which in this case is genotoxic. So you want to limit as much as possible your exposure to it. And finally, the information that you can get. In all the other options, the information is mostly quantitative. But in our platform, you can actually see the bacteria and how they are affected by this specific substance. So for that, we have this specific setup. And here, what I want to emphasize is that the chip is printed in-house, and it's not in the microscopic range, it's more in the millimetric range. That means that what is microfluidic here is the environment. You don't need to have a fully microfluidic setup, but you can actually adapt little by little by the different environment in order to already obtain a level of some of these advantages that we have discussed about microfluidics. So for that, we developed a full protocol that starts with the chip fabrication and filling it with agar, and then sealing, and then it's when the microfluidic environment takes part. And actually, even though you can also make it manually, you can benefit a lot from automating the full sequence. And this can include very complex steps like mixing, perfusing through the agar wells, and then drying in order to be able to incubate it. And then further on the line, you can even include some steps like staining by profiting these capabilities, then to finally observe what's happening. So this is just a little bit of a view of the chip. So as you can see here, you have the 3 different wells, and there's going to be like the mixing chambers that's for mixing. and also the reservoirs that are for the beginning, the bacterial suspension, and the waste. And also what I want to say with this is that everything is contained on the chip, so you again are limiting your exposure to the bacterial strain, that in this case is a pathogen, Salmonella, and also the reagents that might be genotoxic. So for that, we develop a full automated system in which once you have the loaded chip, you can just load one by one the different reagents that you need with the bacterial suspension, and then flow them in a controlled way, line by line, separately and independently, until they are, for example, the bacteria are sedimented and you're sure that they are there. And then you can, again, one by one, dry them and dispose of the waste in a safe way. After that, it's incubated, and then you can observe the results that are here. So what we're doing after staining with, in this case, we're using life or death staining with Cyto24. So what we want to do is evaluate how much bacteria proliferate according to the intensity of fluorescence that we're perceiving. So for example, in the case of water, our negative control, we have a very low fluorescence. That means some mutations were induced, still happens, but it's very low in comparison, for example, of sodium azide, that is our positive control. There's going to be a high intensity, mean intensity of the bacteria because there's a high proliferation. And in this case, our test subject is vitamin C. We can see that indeed it's also quite genotoxic. So we can benefit from framing them between the two comparisons, the two controls. And then I'm going to go to another project that is the study of biofilms, also using a microfluidic environment, microfluidic setup. So what is a biofilm? A biofilm is just the aggregation of bacteria in communities in which they come in contact with the interface between two mediums, and they produce a matrix in which they are embedded that grants them an advantage for survival in a specific hostile environment. And these, for example, can take the form mostly of antibiotics that will come back later. So yeah, the usual life cycle is that free-swimming bacteria come in contact with this surface, and they will attach to it. And after attachment is done, they will start producing this matrix. This matrix will already give them advantages, and then you will have a mature biofilm where different bacteria are actually specialized in different tasks that benefit the whole community rather than the individual. Then when the conditions change, the biofilm will disperse, and this bacteria will try to colonize other environments. So we're starting with the simplest case, that it's using a commercially available straight channel and trying to grow biofilms in a straight channel. For that, we're using this type of setup that looks quite similar to the one that Evelyn showed before. The main focus here is be able to control the flow, because we're going to compare the difference between having no flow and having flow when growing biofilms, how that it can be affected. So for that, what we obtain is that when there's no flow, biofilms grow bigger, but also with a looser structure, as opposed to when there is flow, where you can see a more compact structure and also a much slower growth over time, as we can also see in the quantitative information that we obtain. But we wanted to go a step further and try to make more of a complex case, and this is by growing biofilms in a droplet microfluidics setup. For that, again, we're using a commercially available microfluidic chip. And we're going to have a formulation using first fluorinated oil, which is a high oxygen permeable oil. We want to keep our conditions aerobic. And then as dispersed phase, we're going to use the usual growth medium of our subject, that is Bacillus subtilis, along with some promoters that we want to see whether or not they help biofilm formation or they inhibit it. And then once we have these droplets formed with encapsulated bacteria, what we want to do is trap them and make them stable over time. And we're going to do that through floating, that it's basically at the top of your microfluidic chip, there's going to be a dome. And in this case, we're using an oil that is denser than water. So once we have a droplet and it comes through this dome, it will actually float and be trapped and stable over time. So for that, we have this setup in which I want to focus now on the multi-injection valve, because this will allow us to enhance our parallelization quite further. So as you can see here, we have different processes. So the multi-injection valve will actually help us with the first step, that it's sequencing. So as you can see here, we're going to have 2,261 traps It's a lot of traps. And we don't want to perform all the time the same experiment in the same chip. So what we're going to do is profit to make a sequence of different sample tests of which they will yield different types of droplets. That will make that in your experiment, you will have different types of droplets side by side, and you can compare how they are developing in real time. Then once they get to the flow focusing geometry, the main feature here is that flow focusing geometry allows for monodispersed droplets. So there's going to be a very narrow size distribution of the droplets. That is what we need for the traps, because ideally what we want during the trapping is that there's only one droplet per trap and that they remain stable. So another droplet cannot actually affect a droplet that is already stable. And as you can see here, we can actually produce different types of droplets, and in this case represented by the different color dyes, one side by side and in the same chip. So what we want to do when we encapsulate bacteria is to trap these droplets and see how the biofilm develops. Given that there's no solid support, the biofilms will drop to the bottom and they will be optically accessible at all times, and we can see them grow in real time. And this is how we obtain this time lapse, in which first we can see the planktonic free-swimming bacteria, and then progressively come together. They make a true biofilm, and then they continue until the dispersal, the conditions change, they don't want to be in the biofilm anymore, and some of the cells are released until there's barely any biofilm. But also we want to obtain some quantitative information, not only qualitative. For that, we analyze biofilm information over time, and profiting the sequences that we made, we try to compare different conditions. In this case, we benchmark this platform by trying to see if they could see the effect of glycerol, that it's a well-known biofilm promoter for Bacillus subtilis. So what we can see here is that under the presence of glycerol, biofilms grow faster and also to a higher size, a bigger size, before they start their dispersal. But then we also wanted to apply this, instead of having continuous measurements over time for a time lapse, we also wanted to see the capabilities of this platform to be useful for single time measurements. So for that, we're going to do we did an antibiotic screening, and we measured just after 8 hours of culture, and we wanted to compare what happened. So for that, we're profiting from the fact that this bacteria, it's a specific strain that is autofluorescent. So that means that when they are free swimming, they're going to be fluorescent in a different wavelength than when they are doing endospores. And endospores, I want to emphasize that they are a consequence of biofilm formation. So that means that the more endospores we have, the more relevant, the bigger the biofilm that it was. So for that, what we did first is just make a measurement and try to quantify of all the droplets that were produced at different concentrations of antibiotic, which percentage of those were actually developing into a community of bacteria, which we denominated as proliferated droplets. So here we can see that the more antibiotic we put, the less droplets are going to be populated, which makes sense, given that the minimum inhibitory concentration, that means to stop the growth of the bacteria, for this strain of Bacillus subtilis using pen strep is going to be 4 microliters per milliliter. But then of those proliferated droplets, we want to study the different life states. So for the first one, if we try to study and we try to focus on the free-swimming bacteria, we're going to see that it looks quite similar. So actually, the more antibiotic you put, the least free-swimming bacteria, or the less intense is going to be the fluorescence signal from the droplets. But when we're trying to look at the endospores, it's going to be different. And actually, we're going to get that if you use antibiotics in a minimum inhibitory concentration, you're going to be actually triggering and threatening the bacteria, which is going to try to make a biofilm in order to defend against the environment. So that's why actually it's very dangerous to use antibiotics in a concentration that is not the right one. Because once they form a biofilm, this bacteria actually can have a tolerance of 1,000 times the usual concentration. What would be the minimum inhibitory concentration can be quite higher. So these bacteria can actually survive in high concentrations of antibiotic, and this development over time can actually help them develop resistance. So with all these capabilities that we have, now I want to give a little bit of a broad view of what are the applications that we can have. And sometimes these are applications that we want to get, something that we can get out of it, and sometimes they're detrimental. So things that we want to kind of avoid. And these have a lot of impacts. So for example, first I'm going to start with the microbial dark matter, that it's all of the microbes that are actually uncultured to this day because we cannot replicate the soil properly. This kind of getting us from achieving any of these other applications. So for that, we have, for example, in soil, the development of fertilizers and biopesticides because of the communication with plants, and specifically also bioremediation, because these synergistic paths can actually help a lot in achieving the degradation of pollutants or their neutralization. We're talking more about biomineralization. But then there's also other things that are relevant to us, like water treatment, and for example, fermentation in the food industry, probiotics, or antibiotic discovery. In the case then of detrimental part, we have, for example, personal hygiene, and also biofouling, bacterial contamination, and also for the sterilization of environments, or also antibiotic resistance, because bacteria are actually the main reservoir for discovering new antibiotics that the bacteria have not been continuously exposed, and are, for example, the origins of chronic infections or in implants, etc. And with this overview, then thank you very much, and I give the word to Olivier.
Okay. Right, do we have... Oh, yeah, we have control back.
Yes.
Great. So thank you very much, Jesus. Can we... Okay. Thank you very much, Jesus, for this very nice presentation. And good morning, everyone. I'm very pleased to present the last part of our webinar dedicated to innovation within microfluidics, and more specifically, how in the MIC we envision innovation in microfluidics. So basically, I would like to get back first on what is the MIC, what is our DNA, to understand why innovation is so important to us. So we are a player in innovation for microfluidics, and we tackle many different fields, but especially we apply innovation to biology. And this started from a vision, which is that microfluidic technologies are a cornerstone of the ongoing biotech revolution. And this revolution can enable to push further the limits of science and technologies in life sciences, for instance. And the mission that we gave us is to enable this technology, so microfluidics, to the broader audience and to the ones who are really doing the science, so you, the scientists, basically. Over the course of the presentations, we showed many examples why microfluidics would be beneficial for biology, soil studies, or ecotoxicity studies. But then you may ask, why aren't biologists already using microfluidics? And that's a completely fair question. This study from 2022, published by the University of Oslo, surveyed a population of researchers who are not familiar with microfluidics in the specific field of organ on chip, and asked them why they would not already use microfluidics. And almost half of them answered that microfluidics is or sounds too complicated to them. And it's true that if you're not taking the proper steps, a microfluidic system can easily become as intricate as a network of vines in a rainforest. So basically what we're trying to do here at the MIC is to build compact prototypes and user-friendly platforms to really get over this step and just allow you to do your research. So to illustrate this, I'd like to review a couple of prototypes and platforms that we are developing and which we think can be of use in the context of soil study or ecotoxicology studies. So this first one is a system which allows you to parallelize your experiments. So Jesus mentioned one way of parallelizing, which is droplet microfluidics. This one, basically in each of these small green units, you can basically run a single experiment, and then you can just plug these units one to another and run simultaneous experiments. And well, even though what it does is not impressive. What we wanted to highlight here is a focus on the user friendliness of the system, so that it's very easy to use. So, for instance, we have self-assembling modules. We have reduced tubing, which is always a nightmare in microfluidics. And that's very the spirit of what we're trying to do. On a more practical part, Evelyn mentioned that we can do experiments in hypoxia and anoxia conditions, so specific conditions which do not use only air as the atmosphere. So we developed this tabletop pressure controller with multiple channels, which is ideal to work with gases different other than air. So nitrogen, carbon dioxide, or any other gas mixture to work in, for instance, hypoxia or anoxia conditions. So that's to, let's say, power your circuit with air, but then you need also to control your environment. So we are also working on integration of optical sensors, which are able to sense and control the quantity of a specific dissolved gas. So we are mostly considering optical sensors because they are a non-invasive method of probing this environmental parameter, which is ideal for biological applications. And as you can see here, we have been able to maintain in some part of the system a oxygen concentration lower than the atmospheric one in a stable manner, so working in hypoxia conditions, which can be very interesting in soil. You can also think of working with a pH instead of oxygen, which can be sensed likewise. Talking again about environmental control, temperature is also a key parameter which you might want to play with. To do so, we're developing also this thermal chamber, which is a kind of incubator, if you want, which allows you to work at a stable temperature between room temperature and 70 degrees Celsius. And what is really nice is that it is compatible with microscopy, so you can perform live imaging within a controlled temperature conditions. Yeah, which is quite handy. So here I focused mostly on prototypes, which means very early stages instruments, if you want. But we all know that not two biological experiments are the same. So we also need to think about things that are specific. And that's another focus that we have here at the MIC, which is tailoring platforms which are specific to applications of our partners. So I'll go through two examples, which are for now applied to cell biology, but you can easily think of ways to applying it to soil studies, so either ecotoxicology or microbiology. The first one is used to assess the cardiotoxicity of chemicals, and it consists in this black platform here. In each of these line here is also an independent loop, so it's an alternative to the green prototype I showed you. But this platform is completely compatible with the incubator, so you can just put all your culture inside your chip, fill the system and put it in the incubator, and then let it run for as long as you wish. We're also developing more integrated and complex experiments. That's an example of a platform which is used to understand the drug transport mechanisms of compounds. Of compounds from the maternal blood to the fetal blood through a membrane. And this specific platform integrates a lot of different modules. So we have an injection model, a sample collector, temperature control of the chip, like recirculation, you name it. And everything is automated through the software. So that's another important part of our work is, and of also microfluidics, is that you can easily automate your biological experiments and hence increase the reproducibility and the interoperability of your experiments. So I presented all these innovation that we are doing at the MIG, but we are not doing it alone. That would be foolish, because we are only doing the instrument, but the instrument needs to be tailored to the application and to the research that it will be used in. And that's why we are always striving to develop these things in line with our partners' need. So as Evelyn was mentioning, we are involved in a lot of different European projects, spanning over a variety of applications. So we have a huge focus on cell culture, but we are also working on droplet microfluidics, on chemistry, on microbiology, on soil study. So a lot of different applications. And really the idea in these projects are to build a platform tailored to the partner's need, and then with a series of back and forth, improving it over time to really match the needs, the specific needs. And so talking about European project, it's time also to introduce the EDAPHOS project a bit further. So the EDAPHOS project is a project within the Mission Soil program, coordinated by Michel Chalot from UBFC, which will last 4 years. It started in 2023, with a budget of a bit less than 7 billion euros. The objective of this project is to accelerate land rehabilitation and to restore contaminated soil based on innovative nature-based solutions. So to achieve these goals, we are 12 partners from 6 different European countries with very different field of expertise. And the aim is to contribute to a soil deal for Europe. But then what is the overall concept of EDAPHOS? How do we want to achieve this goal? And how can microfluidics help us achieving this ambitious goal? So we built a project on 5 different pillars. The first one is improving the methods to map the sites that you want to remediate, so that you are able to follow easily the status of the remediation, basically, with new technologies. The second one is to find a way to assess, to define a new way of, or a better way to assess the risk based on the triad methodology. The third one is then to demonstrate the effectiveness of the natural-based solution that we'll put in place to effectively remediate the soils. And all of this will be gathered in a set of KPIs and performance evaluation of the method. And last but not least, we think of this project as a completely holistic approach to soil remediation. So it's also important to consider the financial and engineering part of the system. And so within these 5 pillars, we think that microfluidics can be very important in 2 of them specifically, pillar 2 and 3 respectively. To assess the risks, microfluidics can help developing a user-friendly and easy-to-use ecotoxicity assessment tool. We can think, for instance, of the genotoxicity platform that Jesus made. Things that are very user-friendly and, let's say, portable, which is very handy when you're doing soil field studies. We can also use it to culture microorganisms such as nematodes, a very widely spread model, to assess risks, or just to analyze simply the soil samples to get the access to their physical and chemical properties, to filter or extract substances out of it. There are really a lot of possibilities. Also, in demonstrating the effectiveness of the natural-based solution, we think can be of use in helping to optimize remediation strategies. We mentioned that the parallelization and the sheer number of experiments we can do with microfluidics is very appealing. And also then to test the effectiveness of these remediation strategies, we can also co-culture biological models with soils which have been treated with this solution. So there's really a large horizon of things that we can do. So basically, we showed here and underlined during the presentation a part of how the Microfluidic Innovation Center can be involved in European projects. So the obvious ways are through technical work, so developing new prototypes, developing application-centered platforms or flow control setup. But we also think that the aspect of innovation is very important. That's a place where we also have an expertise. So innovation and bringing a technology from the lab bench to the market as to increase the impact of the research we are doing during the European projects. And so that this research goes beyond the scope and the timeline of the time of the project, but has a wider impact in the scientific community and the society as a whole. We can also count on our search engine optimization tool, so SEO, so our website, which is quite informative and really helps us to define the specifications of the platforms of the prototypes and such. So I heavily mentioned that we are working with our partners. That's one part. But since we want the project to have the broader impact as possible, we also need to get in touch with people who are not within the consortium, so what we would call the market. And this website is really a precious tool to do so and to really find the middle point between these two worlds. Unfortunately, that's one thing we cannot do, which is complex chip design. This requires very specific skills that we do not have in-house. But for any other things that is related to microfluidics, do not hesitate to ask us, and we'll be very happy to discuss the matter further. I just put here a list of upcoming calls within the Horizon Europe project, which we think can be synergistic between our skills and the world of soil microbiology and ecotoxicity studies. So if any of them appeals to you or rings a bell, same thing, do not hesitate to contact us. We'll be very happy to discuss that. So finally, we reached the end of our presentation. So over the last 50 minutes, we discussed how microfluidics is a rather new field, which might seem quite complicated at first, but which really is very approachable and that we really encourage you to use to boost your research. So Evelyn showed that you can have a large variety of applications. And what I think we must remember from that is that with microfluidics, you can easily reproduce the experiments that you are used to do on your bench, but you can also discover a whole new horizon of kinds of experiments that are much harder, if not impossible, to do on the bench. And we can think, for instance, as microfluidic as an easy way to check the spatial and the temporal evolution at the same time of a system, and that's very easy to do. Jesus dived a bit further within the technicity of microfluidics and gave an example how you can have like actual research performed with these tools with two different approaches. One with an inflow, milli, or microfluidic system, and the other one with compartmentalized microfluidics, so droplet-based microfluidics. And I think the highlight here is that it showed that with microfluidics, you can have really easy access to automation, replicability, and low consumption of reagent and time within your experiments. And last but not least, we truly believe that innovation is key in enabling microfluidics to the wider audience and to foster research in life sciences. And that's really what we are trying to do to boost the research. And we do that in two ways. The first one, which I discussed extensively, is collaboration through European projects. But also, if any of the prototypes we've presented rings a bell or seems interested to you, we can completely discuss further and see how we can cooperate through a beta testing program to improve them and expand the limits of science. With that, we finally reached the end of the presentation. I strongly encourage you to follow the EDAPHOS project on the social media, so on LinkedIn and X specifically, to follow the latest news on the latest progress on the project. Sorry, and you can also follow the Microfluidics Innovation Center on LinkedIn to stay tuned on the latest prototypes and platforms we are developing here. And with this, I thank you all for your attention.